12. Today we're going to be talking about the changes in the genetic makeup of a population. So this is Unit 4, Area of Study 1. It is the first dot point set out by the speaker. There are 3 key knowledge dot points that you need to know for this particular unit that we're talking about today. We're going to be looking at how alleles change over time within a population. So we're going to be looking at a few specific mutation types: point, frame shift, and block. We're going to be looking at new alleles, chromosomal abnormalities, so when we have trisomy 21, for example, with Down syndrome. We're going to be talking about the bottleneck effect and the founder's effect, so how we look at the genetic drift or the change in whole mass genetics for populations and the factors that can influence these types of things. We're going to be looking at creating new species, how we know when we have a new species, and we're also going to be looking at manipulation of gene pools through selective breeding programs. So things like if you think about the pet dogs that you might have at home, if you're looking at the apples that you're eating, things that you buy from the supermarket, that most of those things go through selective breeding programs. To start off with, we're going to be looking at gene pools and what gene pools actually are. All that a gene pool is, it is the total of all the genes for a population in a particular area. It's nothing too great, it's nothing too difficult. You should be able to remember this. We're talking about the frequencies of various alleles within a population. So you remembering alleles are different versions of a gene. So whether you can roll your tongue or you can't roll your tongue, they are the same gene. They are different versions of that gene. So when we talk about the frequency of that, it's how many of that allele we have for that particular gene in that particular population. So how many people can roll their tongue? So what is the frequency of people that contain that information in their genes versus the people who contain the information to not be able to roll their tongue in the particular area for the particular people as well. So we're looking at individuals within the population who have some but not all of the alleles that are available to them, the complete total amount that make up who we are. So we're looking at getting this information from a range of different sources. There's a whole heap of different ways that you can go about this. Basically, we're looking at surveying populations to get this information. So we do this by collecting blood samples from different animals, putting trackers on them, the whole catch and release programs that run if you're looking at animals out in the wild and those types of things. But we're looking at being able to collect this information so that we can track whole populations, and then we can determine things that might impact on their survivability long term. We're looking at the frequency of these alleles and how they tend to change from one generation to the next generation to the next. If the environment doesn't change, if there's no migration, so things moving out, so animals moving out from the area that they live in, or immigration, things moving into the area which they didn't live in, but the population we're looking at does live there. And if there's no mutation, if there is no gene change in that particular organism. So we're looking at how genes are changing in a gene pool, so for a population over a period of time. Another important thing to note about when we're talking about these changes in genetic information is mutation. A mutation is anything that is a permanent change. So we're looking at changing those nucleotide bases in DNA. So changing a T to a G, an A to a C, any one of those little changes, that is a mutation. And we're looking at these things occurring spontaneously. They just happen. It can be a lot of different factors that influence that. We're talking about things like UV radiation or any of those types of things. You might have heard to them being referred to as a mutagen, something that causes a mutation in an organism. And every living thing is susceptible to mutations throughout their lifetime. It's not always known exactly what's going to cause these changes or exactly if the change is going to be good or bad. Some mutations help organisms to live, while others can be detrimental to their survival. The effect of them really varies depending on how many bases are affected as well. So if we're talking about it being a whole chromosome, then we're looking at a wide range of changes that are impacting on this particular organism versus 1 or 2 bases on a gene. It really depends as well, because we've also spoken about our coding and non-coding regions of genes as well. If the mutation occurs in a non-coding region, then that mutation won't impact on that organism's survivability, but it could impact on other things for other organisms. The mutations in the DNA can be caused by mistakes to DNA replication. In units 1 and 2, we looked at DNA replication, so how we make multiple copies when we talked about meiosis and mitosis. So when we're replicating genes to make new cells, whether that is for reproduction or not, we can look at having things that go wrong with this process, the incorrect base being inserted, and that's a way that we can get a mutation as well. But then, as I spoke about just before, we have these mutagens, these factors that are influencing this mutation rate that can cause it to happen faster than what would normally occur. So radiation, UV, temperatures, chemicals in our surroundings, they're all causing mutations to the genes that we're looking at. And just remember, they can be very, very small, or they can be quite large as well. There are 3 types of mutations that you need to be aware of. The first one is a point mutation. The second type is a frame shift, and the third is a block mutation. The first one, the point mutation, can be something like a substitution. As you can see in the example here, we've changed the G to a C. So we're just subbing it out or swapping one for another. In the next line here, we have an insertion. So this G has been inserted, which shifts all the bases after it down one. So if you're looking at your 3 base pairs for your coding areas that you're looking at, that can shift all of your triplets that you're going to be reading from. So insertion changes more than just one particular gene, where a substitution could only change maybe one amino acid that you're coding for. And then the last part of a point mutation is deletion. So in the example at the bottom here, you can see that the T has been deleted out of the DNA code. Very similar to insertion, if you take out a gene, all the triplets after that get shifted forward by one base. So again, you're changing your triplet codons, so you're changing potentially the amino acids that follow it. The next ones are the point mutations. Again, this is just a little summary here for you. We've spoken about deletion, insertion, substitution. You have silent mutations down here. These are the ones where something changes, but nothing really changes in the long run. So whatever has swapped still creates the same amino acid, still makes the same protein. You've got the other examples here of the frame shift, which is where you shift how the sequence is read, so your deletion and insertion. You've got nonsense changes, where you're changing your chain determining codons, and then you've got missense, which is looking at a different amino acid resulting from that protein as well. So this is a really great summary slide for you here. Just have a good read through this one. Our next type are block mutations. So we're looking at segments or sections of chromosomes rather than just the singular bases that we were talking about with the point mutations. So duplications is what it sounds like. You're duplicating or copying, having many copies of the same section of a chromosome. So you're looking at expressing a gene potentially more than once. Inversions are exactly that. The genes in that chromosome are then taken out, flipped around, and put back in. So they're in the inverse order to what they were originally in. Deletion, you take that section of the chromosome and you get rid of it. You delete it, it doesn't exist anymore. Insertions are when you can take a part of one chromosome and maybe add it into another one, or insert a whole new chromosome section that randomly got duplicated altogether. And translocation are where we can look at swapping segments of chromosomes with each other as well. Here's a nice little summary for you. In the first one here, you can see that we've got 2 sections of what was on the left in the one on the right. So that's a duplication, multiple copies. Inversion is the next one. It's just flipped upside down. Deletion, it's gone. Insertion, we've taken a particular gene from one chromosome and we've put it into another. And then the same thing goes at the end here with translocation as well. The mutation that we're going to look at is the whole chromosome mutation, and this is where you get your aneuploidy or your polyploidy. We're talking about deleting whole chromosomes or adding in whole chromosomes. So the important part to remember here is that you're going to be having more or less than your normal chromosomal number. For example, having 45 or 47 chromosomes in a human, where our normal chromosomal number is 46. And then polyploidy is having more than the 2 homologous sets of chromosomes as well. So polyploidy is having multiple sets of chromosomes, where your aneuploidy is your deletion or addition of singular chromosomes. So this is the example that we have here. So in this aneuploidy example, you can see that for chromosome 21, there is 3 of them, where the rest of them all have the 2. So we've just got that extra chromosome being added in, and you should all know this one. It is a very common chromosomal mutation. This one here is Down syndrome. And on the right, you can see polyploidy. So you have the haploid, which is the half set, the diploid, which is the full chromosomal set number that we should all be having. And then you can also see the polyploidy examples here of the triploid, where you're looking at having 3 sets of all chromosomes, or the tetraploid, where you have 4 sets of all chromosomes. So both of those are whole chromosomal abnormality types. Then we're going to start looking at the environmental selection pressures on phenotypes. The first thing to remember here is what a phenotype actually is. So your phenotype, guys, is the expression of the genes that you are seeing. So we're talking about what eye color someone has, what hair type or hair color, how tall, how short, freckly, skin color, all of those types of things. They are all phenotypes. They are all expressions of the genes. Do not get confused with genotype. Genotype is genes. Phenotype is the expression of the genes. So the first example that we're going to be looking at in terms of selection on a phenotype is natural selection. You've probably heard about this one. You've probably seen 50 million memes about natural selection. My favorite one is when the guy's in the pool and he has the speaker with all the chords. It's like natural selection at its best. Let's just take all the little tags off things and see what happens. That's not really a great example of natural selection. Natural selection is looking at a gradual change in the frequency of genes over an extended period of time. So Charles Darwin is the one who proposed this theory, and he has a piece of work called The Origin of Species, and he's talking about the fittest one being able to survive. And when he's talking about fittest, he isn't talking about the best looking one necessarily. It is which organism has the best characteristics to be able to survive in a particular area. So you can think of this as survival of the fittest. Which organism has the best characteristics to live? It could be that their beak is pointy, so that they're better able to get bugs that live in trees. It could be that their claws are sharper, so they're able to hold on to things better. It's which characteristics are best adapted to the environment in which you find that particular organism. Biological fitness is a measure of the relative proportion of the fertile offspring that's left by an individual in the next generation. So if something is able to have children, what is the proportion of those that are then able to have children themselves? So it's the relative proportion of fertile offspring that are left in a generation. Because when we're talking about something being fit, it's great, but it has to be able to pass on those characteristics, and that organism needs to be able to survive by itself as well. The next part of that we're looking at here is the inherited characteristics and how an individual can survive in its environment which it lives, and that is called an adaptation. So anything that helps an organism to live is an adaptation. So think giraffes with longer necks. Gradually over time, the ones with the shorter necks wouldn't have been able to survive. So the ones with the longer necks have that adaptation. It is a characteristic that enables them to survive and therefore be better able to reproduce. If you can't reach the food to stay alive, then you're probably not going to be able to make children and be able to pass on your genetic information either. So the key points here with natural selection is that it is a gradual change over an extended period of time. We're looking at adaptation, changes that are being able to be passed from one generation to the next that help them to be able to survive in their environment as well. That is natural selection. This is an example that we have here. So on the left, you can see that we've got some creamy colored mice and some darker colored mice. When the bird flies overhead, it's only going to see the lighter colored mice. It might spot maybe some movements of the darker ones, but it's hard for them to see. So what's going to happen there is that the bird is going to eat more of the tan colored mice than the black colored mice. If there are more dark mice that survive, then they're better able to be able to pass on their genetic information to the next generation. Some tan mice will live, some tan mice will be able to reproduce, and they too will pass on their genetic information. It's just that more of the darker mice were able to live, so therefore there is more of their offspring in the next generation. As you can see in the last picture on the right, there is more black or dark mice surviving than there are the tan mice. This is a prime example of natural selection. It's a characteristic that suits this environment better. And if it was to change, say the ground isn't dark anymore and it changes color to be more tan, then you would expect an increase in the population of the tan mice while the black mice start to die off. It all just depends on what's happening in the environment around them. So the results for natural selection, over a few generations of natural selection, we can look at this variant or the variations in these phenotypes to be better. So you're looking at that selective advantage becoming more common. So think the darker mice over a longer period of time. It's an advantage to be born a darker mice than it is a tan mice because you're more likely to be able to survive. Over a long period of time, you can look at natural selection creating subspecies. So if the two mice start to break away, maybe they don't live in the same environment anymore, the tan mice might have their own little adaptations to help them do things. Maybe they run faster so they're better able to get away from the birds, and that's how they've been able to survive. If those mutations continue to happen, eventually they can become two separate species when they can no longer interbreed with each other. So natural selection can result in subspecies, which is kind of like, think your dogs, you've got different breeds, or entire new species altogether. So gene flow is how alleles move within a population and within areas. So we're looking at alleles coming into an area, but also alleles being removed from an area. So immigration is entering a new environment. So one population is generally balanced by the leaving of an old environment to a new one. So immigration moving in, emigration is moving out. You do need to know the differences between these two things, when things move in versus when things are leaving. So if we talk about the population of Australia, for example, we recently just hit 25 million, which is a lot of people, but we've also got a large number of people who are leaving Australia. We frequently hear about people coming here on visas and living and working here and creating their life here. But at the same time, there are many, many people who choose to leave Australia and live in other areas. So if people are entering Australia, you get the increase in the frequency of alleles. But if they are leaving, you get the decrease in the frequency of alleles. So you can look at if you've got a particular species of fish that live in an area and some of them decide to leave and go live somewhere else, then your original gene pool that you were talking about now has less fish in it, so its allele frequency will be less, versus if something else moves into the area, then their gene frequency might increase as well. So you need to know the difference between immigration and emigration. The thing we're going to talk about is genetic drift. And these are changes in frequencies of a population, but it's due to chance events. It's not necessarily something that is planned, but there are two specific examples that we're going to be talking about. We're going to be looking at the founder's effect and the bottleneck effect. Now, these can be by choice, so organisms may choose to leave and start a whole new population group, but it can also be things like natural disasters. You need to keep in mind that the smaller a population is, the greater the effect of the founder's effect and the bottleneck is going to have on their allele frequencies. If a population is very large, then it might not necessarily have as big of an impact as it would with that smaller population. So we're going to start off with the founder's effect. The founder's effect is when a few individuals choose to leave a population group and start their own little area. So examples of this that you may be aware of are, think your breeding programs in zoos. They would take one or two organisms and then they're going to look at breeding within that population group, and generally they send like a panda bear, for example, from one zoo to another so that they can look at sharing that genetic information between the zoos. But that is a breeding program. It is a small population of all the pandas that can be found in the world, and so their allele frequencies don't necessarily align with the bigger allele frequencies in the natural world, in the world outside of the zoos that these, the panda bears live in. So if the numbers of the individuals are low, then alleles for certain traits are going to have less variation within them. You have the higher chance of small less popular genes being seen. So there's a greater chance for some of your recessive conditions being expressed, particularly if those who are leaving are known to each other. Maybe they've lived in the same area for quite a while. You can look at having those types of things together. And we're looking at over time, there is an increase in the variation between the old population and the new population. It does take quite a while, but this is one of the things that can cause subspecies or speciation. The creation of whole new species can arise from this one as well. It is due to the selection pressures from their environment. So if an organism chooses to leave one particular area and then the food source in the new area that they live in is markedly different to the food source in the old area that they lived in, then they're going to have favorable traits being passed on from one generation to the next that suit the new food source that they're found. So we're talking about those little changes, but they are consistent over time. So the favoritism of a particular beak type or claw type or gill shape in fish, or whether or not their scales are sparkly, all that kind of stuff. Over time, the creation of those two groups can develop into separate species. So if we look at this example here, we're talking about a small group. In this case, it's green and orange, making a founding population and in a different area. So what that means is this founding population actually is missing a large section of the genetic information that the other group had. So they didn't have any red, they didn't have any purple, and they've separated out, so their genes are being passed on more heavily. The example that you're looking at now on the screen comes from the Amish community. They are a small subset of people who chose to break away from the traditional society that we live in, and these people tend to have extra digits. It is a very common trait within the Amish community as well. So that's that expression of genes being more pronounced in a particular group because the gene flow in and out of that population group, in and out of that founding little population, isn't as great as what it is in other areas. So this is where you tend to get particular traits being more common within that subset than they are with the rest of the community. The next example that we're talking about is a bottleneck. Now this is an intense selection pressure. We're looking at a larger population size and then something drastically cutting the amount of people or the amount of an organism that lives in a particular area. So this is an extreme case of genetic drift. It is a catastrophic decline in the population size. You're going from maybe thousands to as few as hundreds, maybe even less. It depends on exactly what's happening in this bottleneck. What happens is that the allele frequency of the organisms that survive might not be the same percentage frequency as what the original population had. So the surviving members do not always represent the whole population of what it was before the event occurred. So again, you can get this smaller population that results from the bottleneck effect passing on a smaller allele frequency, and then you can then have the lesser pronounced traits being more common in that population group. So we've got a limited genetic variation. There's not as many organisms there for them to be sharing their genetic information with. So then some of those traits are going to become more popular. If you have a look here, the former population on the left has blue circles, it has green triangles, it has a larger variation. A bottleneck effect severely reduces the population. So something happens, whether it's an earthquake that kills off quite a number of the animals, or it could be the tsunami, or it could be flooding, it could be wildfires. Any one of those things can kill off a large population group, leaving you with this smaller subset of what was originally there. The genetic information that can be passed on is only from those that survive. So the new population don't have the same genetic variations and frequencies as the original population had. This is why the bottleneck is that extreme example of genetic drift and those genetic changes. The next thing that we're going to be looking at is evolution that occurs from mutations, so those tiny little changes, and how over time we get evolution, how evolution actually happens. There are a few things that I want to point out here just before we dive into it too deeply. If your religious or cultural beliefs do not align with the theory of evolution, keep in mind that it is just a theory, but when you are doing your exams for VCA, it's quite important that your views that are expressed need to align with what the study design is asking from you. So in this exam, you will be talking about evolution, and you do need to talk about it from the scientific standpoint, even if your personal beliefs don't align with that. So evolution occurs when a gene pool permanently changes. So you get that change in their genetic information, which gets passed on from one generation to the next. We're looking at forming subspecies from what was pre-existing. So you can get different versions of the same organism, or whole new organisms altogether. That the idea of speciation can come into that. So evolution of a species occurs by natural selection, and it takes place over time. So it takes a fair amount of time for this to happen, and the only way that it's going to be successful is if you have isolation. If the organisms can still get to each other and have interbreeding, then it's highly unlikely that you're going to be creating new species or getting this marked change over time. To get the new species, the organisms need to be separate so that they can't get to each other. If they can't get to each other, then you don't have gene flow between the population groups. So that would mean that their allele frequencies are going to be less or high depending on whichever trait you are looking at, so that the particular gene, the particular allele, can't be passed from one generation to the other. The birds at the bottom, they are examples of finches, and we're looking at insect-eating ones on the left here. You've got your ground finches over here. So there's lots of different versions of finches, and you can see that their beak shapes are just slightly different to each other based on the food types that they are eating. So different beaks are suited to the different food sources that are available to them. Evolution is the creation of a subspecies. In order for things to evolve, they need to change from what they were before and become something entirely new, while what they were before is still hanging around. So we're looking at members of one population being isolated from other populations, which is what we've already spoken about today. But we're looking at those different selection pressures or agents due to the environmental conditions of where they are, creating these subspecies. So you're looking at genetic drift on a larger scale, that more long-term type stuff. Different alleles being created as a result of mutation. So the separation of the species, a mutation affecting one population that doesn't affect the other population. And then over time, you can look at the creation of a subspecies or races within that. So if you think about dogs, for example, there's a whole different range of dogs, but they're all still dogs. It's just that they are different subspecies. They can still interbreed with each other, which is an important part of subspecies, whereas if you're creating a whole new species, then you don't get that interbreeding. So subspecies are just categories of animal where they're still genetically similar enough to be classed as the same species without being two different species yet. So speciation is the next step. It's where we start to get the whole new species. So one population divides into two groups. So you're looking at two daughter populations here. This is what's up in here. And they're not necessarily of the same size. So your original population begins to divide. So you have that selection pressure here, whether it is a foundation effect or a bottleneck or whatever it is that causes two populations. And then eventually over time, the two population groups will have the different selection pressures to make two different subspecies that are then becoming different species entirely. That's when you get speciation. If they can no longer interbreed, that's the key for speciation to have occurred. They cannot reproduce with each other if they are separate species. So think horse and donkey. They are very clearly different species. You can force the creation of a mule, but biologically out in the world, it won't happen naturally. Lions and tigers, you can make a liger, but those ligers can't reproduce. So they're separate species. They aren't the same species to each other. The only reason why we get these offspring is because we, humans, are interfering in that process. So look at allopatric speciation. It is the creation of those different groups, firstly by geographical isolation. So that's the first thing. You need the two groups separated by geographic isolation. That could be a mountain range forms, or you get an earthquake and there's this big crevasse that they can no longer cross. So we're looking at something extrinsic on the organism, so external to their own choices, that prevents the two from being able to get near each other for mating purposes. And then eventually we're looking at that creation of those new species by the mutations that are occurring into those two separate population groups. So isolation can be distance, so think birds and fish and things that migrate based on weather, or it can be that physical barrier that we were talking about before, earthquakes, volcanoes, continental drift, erosion, all those types of things can lead to the two different population or more population groups that are formed before we get the subspecies and then speciation that occurs. So this is an example here with some bugs. So we've got some little beetles on the left-hand side. Something occurs, perhaps a new river forms through the area where they lived, and now these bugs aren't able to cross the river to get to each other. So over time, mutation, natural selection impacts on both those population groups separately, and when they are brought back together, so say there is a drought and now that river doesn't exist, and the two populations come back together, except they are not the same species anymore, they're two separate species, and so they're not able to share their genetic information with each other, and that is what makes them their separate species. So what we're going to finish up with is selective breeding, and this is where humans start to be involved in the process of species breeding and looking at creating their offspring. It is an artificial process, so it's the human intervention into what is already occurring naturally, and this might be animals breeding. So if you think about the pugs that we have down the bottom here, over time humans have bred pugs to have the shorter nose because they were bred for looks, and that's what people wanted, even though it's created a lot of respiratory issues in these animals. And the same thing happens with the dachshunds, so the sausage dogs, we're breeding them to be longer and longer, even though that creates back problems for those particular dogs. So human intervention there is one for a particular characteristic, so the phenotype that we like. But very similar things happen with foods that we eat as well. So you've got the example with the carrots on the right hand side. There is a story around the Netherlands and carrots being more commonly produced there way back when. And so because orange was their national color, people would only start to continue to breed the orange carrots. So therefore the other carrots were starting to not be as common. So we were breeding only for a particular type and characteristic and color of carrot. And that's why they're the ones that you're most commonly finding Coles and Woolies at the moment. The same thing happens with corn and also with the animal products that we are eating. We're selectively breeding corn so that we're able to get more product from it, and we're selectively breeding animals so that we're able to get more food and more nutrients for us without necessarily taking into consideration the impacts that it has on them. So what we're going to do is we're going to delve into corn a little bit deeper today. So we're looking at breeding for that particular phenotypic outcome. It can be for humans, we're looking at things like flavor as well as product. So this is on the left here. This is what corn originally looked like. And then we're looking at which ones we're looking to breed. So what we've done is we've bred the bottom line of the corn that we were looking at. So those that were creating the larger corn kernels so that we were able to get more product from it, while not breeding the top ones there where we're not quite getting as much product as what we had previously. So we're looking at not necessarily manipulating their genes, which is what we were talking about in the last topics we were covering, rather favoring a particular phenotypic outcome. If we know that something has this phenotype and we can play off that and continue that crossbreeding for a particular phenotype, then we can get an outcome that's more favorable for us as humans to help us stay alive. So it's not as invasive of a gene manipulation, but it is a form of a genetically modified organism, and it's something to just keep in mind. So that's it for this area of study, this little chunk. Hopefully this will help you guys to make your summary cards and everything else. Good luck.